Welcome to our backyard. This is the Backyard Philosophy Podcast. We are two friends having a discussion after everyone else has passed out or gone to bed. Grab a drink and listen as we discuss everything from automation, space exploration, and why the meaning of life is 42.
We've talked about it in human experimentation and animal experimentation and in bringing back extinct species. And it seems like, Nick, to be honest with you, comes up in just about every episode. It's a keystone in all these circles. CRISPR. Will it be the technology that saves us or the technology that dooms us? But before we get into it, Nick, how are you doing and what are you drinking?
I am doing, well, I'm under the weather if I don't sound the same, but still drinking some Dead Guy Ale as per usual. What about you, Mike? What are you drinking?
Well, I'm drinking something that might mutate me. I'm drinking some whiskey from Cleveland. So we'll see how that one goes. Some Cleveland Underground. I think it's from Cleveland. If not, their name's very misleading. But
so underground, it's not even in Cleveland.
Well, does anything want to be in Cleveland?
Not most major sports teams.
Oh, you with a sports joke. Was not expecting that one. And Nick, once again, this I hate you because there's a lot of scientific names and my English no good.
So Mike's going to continue the rest of this podcast in Spanish because apparently he's better at some other language, just not his own.
No, I'm at the back end of the bell curve when it comes to language skills, but some base knowledge before we get deep into the podcast. DNA. Nick, a little 8th grade chemistry and biology. Do you remember what DNA stands for?
Deoxyribonucleic acid?
Oh, fuck me. What about RNA? Was not expecting that answer.
Something with an R nucleic acid?
Yeah, ribonucleic acid.
Fuck.
God damn, I was gonna grill you.
I knew not to look that up.
Shit. What about, can you tell the difference between DNA and RNA?
Like if there's the letters, like if I'm looking at them, or what is the difference in function?
Let's start with
because the DNA
What's...
has 2 strands, right? RNA only has 1 strand.
Yes, most often RNA only has 1 strand. And wow, Nick, you're on the ball today.
Yeah, I took biology in high school, Mike.
Fair enough. Well, Nick, since you're so good at remembering from high school stuff, technically this did come out or was announced publicly on. Actually, this is all technically before even we went to high school. Do you know what CRISPR stands for?
Oh man!
As you quickly type it into Google, I'll give you a hint.
It's
Nick.
on the tip of my tongue, but I'm not going to be able to pull it out.
Okay. Well, there's a joke there. It is CRISPR stands for clustered, regularly interspaced, short, palindromic repeats.
Just kidding. It was not on the tip of my tongue.
CRISPR, CRISPR. And I think the best way to start about CRISPR is maybe start from the beginning. And in 1987, a Japanese student by the name of Yoshizuma Ishuno found a repeating section of DNA in bacteria. But with technology being slower back then, sequencing the DNA would be put on the back burner. You gotta imagine, this is late 80s. Computers are a luxury, not everywhere.
And not good.
Yeah, well.
I mean, not good by our standards.
Then, in the early 1990s, a Spanish student, Francisco Moca, also noticed a repeating sequence in the same DNA. Fast forward to 2003, DNA sequencing technology vastly improved. Francisco used this advancement technology to investigate the repeating sequence. Now, this all might sound simple to you, like how hard is it to sort through DNA? It's only 4 letters, and they can be on and off, stuff like that. There are 3.2 billion pairs in human DNA, 6.4 billion letters for our 46 chromosomes. Technology was definitely needed. Little sidebar, Nick. I was researching. Do you know what the simplest creature scientists ever created? You know how many genes that creature had?
If I throw out a random number, how close do you think I'll be?
I'll give you a hint. It's between 0 and 1,000.
319.
Not too far off. 473.
I was going to guess 420. That's my first guess.
Damn. Well, 473 seems to be close to, if not the lowest number possible for life, which is quite interesting. But with this new technology and all this DNA, I mean, everything living has DNA, technology was definitely needed. And by 2010, scientists started to understand the repeating process found by the Japanese student in bacteria. This is where the path diverges and gets complicated. In 2011, Jennifer Doudna from Berkeley and Emmanuel Carpentier from the University of Vienna decided together, let's work on this. They work on the sequence of DNA. They decided to work on CRISPR, a relative unknown phenomenon laying in wait. Now, near or at the same time, you have other people also working on CRISPR on different paths. You have Virginijus Skiskisny, a Lithuanian scientist, and you also have a team composed of George Church from Harvard and Feng Zhang from MIT.
I think from what I understand from Doudna getting into CRISPR is they were looking at, I guess what you said, but just expanding on it, they were looking at the RNA. They didn't necessarily know that CRISPR was involved. They were just curious of why this was happening, which led to...
I'll be honest with you, Nick. I actually did not know why they got involved with CRISPR. I just know that they decided to work together. So if you have any more information...
Well, I guess that was the extent of the information, pretty much. It's just, or at least for Jennifer, she, from what I understand, listening to interviews from Walter Isaacson, who wrote the book Codebreakers on her and CRISPR, is that she was researching why they were finding this certain thing, which led them to figure out that CRISPR could insert or was doing gene editing, and then that led to her being involved in CRISPR. She didn't get into CRISPR purposely, kind of accidentally.
Well, I imagine that small caveat of a field. I mean, imagine you don't get into things accidentally, but that's interesting. Just like almost like rediscovering it. Well, anyhow, Emanuela and Jennifer would publish in June 2012. Skiskiny would also publish near a similar time, but his paper was rejected a couple times and wouldn't be published until months later. Also, in 2012, Zhang and Church would publish. A legal battle over the patents would get complicated, but I don't want to get into that just yet. But before I get into the patents, or if we even have time to get into the patents, I want to focus on actual CRISPR, how it is used and how it is made and how it even works. And I think how it works might be a good place to start. So, in bacteria, just all bacteria in general, and yes, Nick, I am generalizing, they are in a constant struggle to live. And one of their biggest enemies, much like humans, are viruses. So, bacteria, in order to fight viruses, produce enzymes. And in this epic battle of fight to the death, and due to the size of the bacteria, they have to be fast and efficient when fighting. Now, this is not seek and destroy. This is more like withstand a siege. So, a virus may enter a bacteria, and the bacteria instantly has to figure out where the virus is, what the sequence of the virus is, and to kill it. This protein, this enzyme, bacteria produces is Cas9, Cas-9. Now, how CRISPR actually works is kind of similar to bacteria of how it removes the virus, though we kind of modified it a little bit. See, it's a combo of Cas9 and an RNA molecule. They are combined, and how it works is Cas9 is sent to a specific genome, and using the RNA, it looks for that match. They attach, Cas9 then cuts the DNA, and the RNA, which we can program, we can make, switches and smooths out the cut. Now, usually, CRISPR is administered through adeno-associated virus vectors, or AAV vectors, because I don't want to say that every time if we ever talk about inputting COVID into people. But the CRISPR goes through the cell, or through the body, depending on how CRISPR is being used. The RNA finds the matching sequence. CRISPR then wraps around. Cas9 cuts, RNA replaces, but CRISPR doesn't just have to cut and replace. You can also change a gene, silence a gene, and promote a gene. All three, which I'll talk about later, but the biggest one everyone probably is thinking of is slicing DNA or silencing DNA.
Yeah, for dum-dums out there like me, I just think of it as like the right click in Excel. You can take out, you cut, copy, paste, insert.
Well, it's exactly like that, but you got to do Control F before you copy and paste. So Control F on Windows, for all you Apple people, switch over to Windows, or actually switch over to Linux. Fuck big companies. Control F to search for a specific word. You find that specific word, you copy, you cut, you paste. That's the layman's analogy, I guess.
Right, because it has to go in like the certain spot. So I think we did a really good explanation combined. It took two of us to get there.
It's a complex aftermath, but I'll be honest with you, the actual process is pretty simple. Though, as a mechanical engineer, we're in biology and chemistry, so all of it hurts my mind. And anytime you modify genetics or organisms, hell, just biology in general, my mind goes,
Well, biology, I enjoy biology more than I enjoy physics, so I'll try and help you out the best I can, Mike.
Cool. I'll try to ruin biology for you. Hey, Nick, do you know what the different types of CRISPR are?
Do you want me to name all of them? Because I am not going to, but I know there's a couple. And they're all essentially the same, but they're all different, and it's going to massively complicate the legality of different people and companies using all of them.
Oh, I wasn't getting to the legality part. I was going to talk just about the different types of CRISPR, mainly class 1 and class 2.
Oh, okay. Because there's like the Cas9, but then there's like a something 13.
Yeah, there's Cas13, and then there's CPF1.
Yes.
Those are like the main ones.
All right, so keep going with 1 and 2.
Okay, so CRISPR is a general term. It's like a car. If you just say a car, it just means a general term. That's kind of CRISPR. When you start getting the specifics of how it's being introduced, what's in CRISPR, then it starts changing to make and model. So a way to think about it is there are two classes in CRISPR. That's just a very generalized statement. If you want to be nitpicky, you could say there's more, but for the most part, there's two classes.
You got your gas and you got your electric.
Yes, that's actually the analogy I was going to use. Perfect. So class 1 is, to me, the electric vehicle. Class 1 CRISPR has multiple subunits. So think of it of like different types of batteries in the car. Some of them are lithium, some are lead acid. That's kind of class 1. Then you have class 2, which is a single large protein. So that's your gasoline car that's just very bare minimum. Like I'm talking like 65 Mustang. Like it's just an engine, a transmission, and if you're lucky, someone actually put exhaust on it. It's pretty just a giant block of metal, but in this case, it's a giant block of RNA and bacteria. Or sorry, I should say enzymes. Now, the main difference between class 1 and class 2 is what you were saying, Nick, is the CANS enzymes, sometimes being CPF1, can cut where CRISPR can't. And then you have Cas13, which can cut RNA instead of DNA. Like we mentioned in the beginning of the podcast, your DNA is your blueprint. Your RNA is your instructions. So your DNA is like the original copy. RNA is like, No, I would say your DNA is like your blueprint and your RNA are the key instructions. So the blueprint is like how to manufacture the furniture. RNA is how to assemble the furniture. I think that's a simple enough analogy that kind of works with that.
Yes.
Before we get too far away from the different types of Cas, I was going to talk about why CRISPR is so important to humanity at the moment.
Yeah, go ahead.
Well, CRISPR has this very unique feature. Like I mentioned, it wasn't just cutting and replacing, but it can turn off and turn on. Based on your lifestyle, different genes constantly turn off. Your body is more like a Christmas tree light than you think. Based if what you're eating, how active you are, your depression, if you're happy, if you're going through high stress, if you're going through low stress, if all those things change your genes. Some turn on, some turn off. But something that you can't help is many diseases are inherited. More than 10,000 diseases are passed on through humans breeding alone. And CRISPR is a new way of gene editing, which requires minimal cost. It actually might be safer than our old methods. Our old methods kind of being brutal. And as we get into talking about it, it might be a implementation where you don't have to catch it early in your disease. You can catch it whenever you want. But it's all complicated. And it's especially complicated because in 2018, in my opinion, a scientist went too far. Ahi Janakin in 2008 altered embryos using CRISPR. He did this. He says he did this because the fathers of these mothers had HIV, so he wanted to make HIV-resistant babies, give them a better life. He was also thinking about the Nobel Prize. I think he kind of did it more in self-interest more than the benefit of humanity. But either way, a pair of twins were born, followed by a different woman, a third child being born, all modified using CRISPR to make the babies HIV resistant. Though the child's names are hidden, which I think is a good thing, we don't know if they're actually HIV resistant. And fun fact, Nick, that scientist was sentenced and in prison until this April, where he was released. And he apparently has been meeting with investors to start a designer baby company.
Well, I feel like that's not too surprising when we all saw the writing on the wall of this coming.
Yeah. If you haven't listened to our human experimentation law, the biggest thing that probably I learned is there's pretty much not a lot of laws on human experimentation. And honestly, in the United States, I feel like the only real law is don't touch embryos without the FDA approval, as bad as that might sound.
And I guess I want to point out that there's a lot of experimentation that does take place with embryos, but most of it, the majority of it, they're not carried to term, right? Like they're just experimented with, and then they just get rid of them. See if
Well, you do get man bear pig.
Well, this is how you get man bear pig.
No, we have man bear pig. If you remember when recording human experimentation, we quite literally have man pig. All we're missing is bear.
I mean, Al Gore warned us.
He was super serial, guys.
After he created the internet, he warned us.
Oh, that's one thing you keep saying that drives me absolutely insane, but I find it absolutely hilarious at the same time.
For those of you who don't know, I think we talked about this on the internet one, but Al Gore helped fund the research that led to the internet, but he had no part in any of that. So, and by say he helped fund it, he used taxpayer dollars, so we helped fund it. So, I mean, we created the internet, but that's not what we're talking about right now.
No, but with the internet, it's definitely helped CRISPR definitely explode. Kind of backtraveling a little bit, speaking with embryos and the different types of CRISPR. So the reason why I mentioned the CPF1 and the Cas13, and then the original CRISPR being Cas9, all those protein enzymes, they cut DNA. They're able to slice through DNA or RNA without causing damage to allow. So it's like Nick, have you ever seen those planes who get refueled in flight?
Yeah, that's what my dad did.
Oh, well, I thought he was artillery for some reason.
Quite the opposite of that.
Yeah, I don't know. I don't know.
Later in his
I don't know
career,
why
he did stuff like similar to that. But
gotcha. That's probably what I was thinking of. But if you think of a fighter pilot getting refueled by a plane in the air, that's kind of how CRISPR works. Your Cas9 is that plane that's going to refuel the jet. And the RNA is the gasoline that fills up that. So your original jet being the fighter pilot, I might be just making this analogy because I have Top Gun on my mind. But CRISPR has the amazing ability to cut like a pair of scissors, an X-Acto blade, without unraveling DNA. And I'm an idiot, and it still utterly shocks me, and my jaw actually drops to when I think about how crazy that is. The blueprint, which is you, is cut, but fast forwarding. Sorry, that was just my amazement of the protein enzymes. But all these different protein enzymes, the Cas13, Cas9, CPF1, there are different tools in the toolbox. They're all able to do things other ones can't. Cas13 cutting RNA rather than DNA, which might be important later on because we might want to modify RNA for a disease but not have it genetically passed on. CPF1, which can cut where CRISPR can't, so certain diseases can't. All this is very important, especially when you're talking about embryos and designer babies. If you know the DNA sequence of both the father and the mother, you know that because those two genes mixing together, that the child's more likely to develop sickle disease or cancer or some other horrible disease, you could be able to turn it off so it never happens before the baby's even born. That's straight out of Gattaca, and that straight hurts my brain, Nick.
Yeah, but that's awesome and scary at the same time.
Well, I think most of the world at the moment is more scared of it. I mean, genetic engineering has been banned and restricted in Australia, Belgium, Brazil, Canada, France, Germany, Israel, Netherlands, and the UK, and probably other countries, not China, but dozens of other countries. So I think people are scared of its amazing power.
Yeah. Do you want to get into that, or do you want to keep talking about what it is?
Well, Nick, morality is high in the question. I want that to everyone to listen to because we'll get to that in a bit. I want to hurt your brain more because it hurts my brain with biology. I want to hurt your brain with patent laws because I somewhat understand those, even though I'm a fool with a capital F. But I kind of understand what's going on.
And I want to also say, as I can understand biology and you can understand math, the legality of things is just terrifying. This is going to get fucked up.
Laws are stupid, Nick. That's all I can say. Laws are fucking stupid. So, all those teams researching, publishing, Church, Zhang, Jennifer, Izu, all those people, they all coming back, and we're traveling back to 2012. So, okay, sticking with 2012, the patent laws for CRISPR, I can't express enough, are a clusterfuck. Duan and Carpentier applied for patents in 2012. In 2013, Zhang and Church applied for patents. MIT and Harvard, were backing Zhang and Church, paid to expedite the patent process, which made their patents approved in April 2014. Duan and Charpentier being like, what the fuck? Berkeley and Vienna, well, they petitioned for patent interference. After many times of being denied and trying to repeal their denial, they finally got to present their case. The U.S. Patent Office, I want to clarify this is U.S., Europe is still up for grabs. The U.S. Patent Office ruled in Harvard and MIT's favor. And the reason why, it might be splitting hairs, but I'll let you decide. So, Jennifer and Emmanuel patents used prokaryotic cells, while Zhang and Church use
prokaryotic and eukaryotic.
There you go. That's why I keep you around. Now, this is an important reason or a dumb reason. The reason why those cells are important is eukaryotic. How do you pronounce it again, Nick?
Eukaryotic.
Eukaryotic cells pretty much means that CRISPR will work on humans. And that's what Zhang and Church showed. Jennifer and Yumel showed prokaryotic cells, which are mainly plants, and that's why there's separate patents. Berkeley and Vienna argued that it's a simple process between one to the other, and they all disagree. So Harvard and MIT disagree that it's a simple comparison, 1-on-1. So it's like Berkeley and Vienna are arguing that all right, we'll switch it to beer. So pretty much they're arguing how to get the beer open. Is it a screw tap, or do you need a bottle opener? That's pretty much what they're arguing about for CRISPR. And Patent Office in the U.S. seems to agree with Harvard and MIT that it is extremely different, a twist off or a bottle opened beer.
And I want to point out that the different labs that are backing these are all receive federal funding. And I know there's private partners, but to me, it seems kind of ridiculous that we're having a bunch of somewhat publicly funded research fighting with each other so much.
Oh, this is all big money. And also, I don't know if the scientists actually get any royalties for this. I think it might just go to all the universities. I could be mistaken.
No, I think it does just go to the universities. I don't think they get.
Then what the fuck? These are 3 out of 4 universities that are all US of A. Why is that not just all free for the United States?
That's what I couldn't find. That's what I was trying to figure out. But I think because this is my inference, is because some of the research was helped funded by private companies. It's supposed to be kind of a joint public-private cooperation so that those private companies can use those patents. But
you know what I would do? I would just have those private companies be free to use. Like they don't have to pay royalties for using them. That's just me.
yeah, that's an easy solution. But I don't really know how I feel, and this is getting a little off topic, about private industry or public research institutes, mostly like UC Berkeley. Harvard receives 70% of their funding comes from the government, but you could still technically say they're private, I guess. About how public institutes helping private industry in this way. I mean, they're supposed to help private industry, they're supposed to help the American people through their research, but it feels more like they're just working for companies at this point than they are people. So it's just kind of confusing, and I don't know. I don't like it. It feels dirty.
To be honest with you, Nick, it feels like universities are contractors.
Yeah, exactly. That's what it feels like. That we pay for.
Honestly, I would just give all 4 of them patents and just be like, you're all right, you're all even across the board. All of you shut the fuck up. That's what I would have done, but not the US Patent Office. Again, Europe is still up for grab.
Well, in the way that there's the International Patent Agency, that once you file a patent with the US, you get a patent internationally in all these countries, but you have to actually file that patent in those countries, but you get a patent pending in all the other countries until you do.
That's actually dependent on what type of patent it is.
Okay.
So if I remember correctly, there are 3 types of patents. There's like the chemistry one, there's intellectual one, and there's like a physical thing one. And I'm not quite sure which one of those this CRISPR falls underneath with those 3 patents. I feel like it falls underneath all 3, but I don't actually know.
Well, I know whatever it falls under for CRISPR, that these patents are technically patent pending in all the other countries, as long as the companies choose to activate them. They can, from what I understand.
Well,
so whichever one that would
be. It doesn't
be.
matter it doesn't matter if it's China, because they're not going to follow patent laws.
That's a whole nother conversation.
Oh, I'll be honest, I'm still just angry. Like, I understand you're trying to help possible HIV babies, but it really feels like dirty. Like, the universities and private institutions feel dirty, but experimenting on pregnant women feels extra dirty to me.
Well, that's a big theme in human experimentation, isn't it?
Oh, yeah, twins and pregnant people. It comes up a lot in human experimentation. But I did find it kind of funny is that Jennifer and Amelia both won together the Nobel Peace Prize in the scientific field, the Nobel Prize, which I feel like if you win the Nobel Prize for it, you kind of deserve the rights to it. But I'm not sure if the Nobel Prize is just a consolation prize.
Yeah, and I don't know how political that is. So with all those awards and prizes, you never know.
It's a lot of money. Yeah, it's definitely got to be political. There's a lot of money on the line. But, kind of sticking with Harvard and Berkeley, Nick, do you know about Berkeley's binding hormone experiment or Harvard's quote-unquote camera experiment?
What's the camera experiment?
So this is all using CRISPR. The acronym for it is CAMERA, I believe, or the project name is CAMERA. I can't remember which. But at Harvard, Dr. David Liu is making CRISPR into a camera. It will be CRISPR, so it can find the specific RNA or DNA. And instead of cutting, it goes to the cell or the area and records whatever it's attached to its entire life, like a time-lapse recording of the cell. Now, this has huge implementations, especially when you're talking about experimental drugs. Like, imagine if I'm giving, I don't know, say you have leukemia, Nick, and I'm giving an experimental drug. Well, I want to know how it actually affects your cells. Well, instead of giving a radioactive isotope, which I can study underneath, machinery, I can just hit you with a biological camera to record your cells. How it actually works, I have no fucking idea. That when you start getting to biology, you start losing me a little bit. And going back to Berkeley, if I understood correctly, are conducting experiments with bonding certain hormones to Cas9. And the reason why this is kind of interesting is if you gave someone CRISPR as a child to cure their disease, or their disease won't be activated until puberty, which is actually very common for a lot of diseases. A lot of stuff doesn't happen until you hit puberty. The Cas9, the CRISPR, won't activate until certain hormones are activated. So not until you hit puberty does the genes activate until CRISPR activates.
So that's like a delayed response.
Yeah, but that's really fucking weird. The camera and the time bomb's not the appropriate word, but comes to my mind, of two different types of CRISPRs really fuck with my brain.
Yeah, that's great. I always thought of it as being a pretty quick thing, like it's one and done, not really a delayed response. But I guess what's the benefit of waiting?
Well, I think if I don't know their reasoning, but if I had to make an educated guess, I imagine the CRISPR would remain dormant and would not activate until your disease pops up. So say, I don't know, say your pituitary gland is enlarged and produces more testosterone. Until your body reaches a certain amount of testosterone, CRISPR won't activate, so it'll shut off that gene that's making you produce so much testosterone. I can see that, but again, a lot of diseases don't happen until puberty. I mean, one of mine didn't happen until puberty. It's when your body's going through those metamorphoses that different things happen. It could be like a safety switch, like don't use CRISPR unless X happens. Well, X happens during puberty, during these hormonal stages. Hit it, or maybe these are all just wild guesses. Maybe during your puberty stages is the best time to change your genes because you're going through a metamorphosis, for lack of better words. So maybe with all those different chemicals, you're able to manipulate the body more or less. Not quite sure. That's just a guess.
I mean, that sounds reasonable. I could understand that. I mean, you'd think I way outside my area of expertise, but you would think doing it initially would be better, but I can also see how, there's certain parts of your genetic code that probably don't get activated until puberty, so I guess you could wait and do it. It seemed, I don't know, in my mind it seems like something that it's cool, but what's the application, I guess?
This is straight out of left field, and this is just all hypothetical. But I'm also imagining for a soldier that you could implement different CRISPR to different parts of his organs. So say if he gets hits in the lung, CRISPR will activate and have a sequence that makes you rebuild lungs. Because we're already changing pigs to humans for organ transplants using CRISPR. So why can't we do it in our own body of like, hey, you didn't stop growing when you're 25 or 26. You actually can continue to still grow. We just have to trick your DNA to say you're actually younger. Or so it's not until that giant stress factor happens, or I don't know, something along those lines. I find it all interesting, and also the camera one I find super fascinating because I don't understand how a protein enzyme can record what the fuck is happening in a cell. I don't understand the science of how a protein records what's happening.
Yeah, I mean, I don't have that answer. You kind of left it open-ended.
Oh, darn.
But could not tell you.
But you know what the main things we are using CRISPR for, Nick, besides,
On humans or?
Just in general.
Plants, I'd guess.
Yeah. We're changing plants, food.
Because I know
food kind of falls into plants.
A company, Corteva, that has a patent on CRISPR for plants. And they're an herbicide company. I'm I work with those guys. Well, obviously not the CRISPR people, but the Corteva reps. I'm familiar with that company. And I know that's going to be a huge, I mean, everyone has seen like the genetically modified crops and it, we did an episode on that if you're interested, but I think that's going to be a bigger application because at least in my world, all the cool stuff goes to ag first and then it comes to everyone else because that's where the money is. And I think there's a lot of money in that. And I think the example that we used is taking I want to say it was for wheat, but taking a rust-resistant protein or a piece of rust resistance from wheat and putting it into, I want to say, chestnuts, roasting on open fire, chestnuts, the American chestnut tree to help it overcome an introduced blight. That's what I'm most familiar with. But I think, and that's just for conservation, chestnut tree is not a huge crop tree, but I think there's a ton of application when it comes to coding plants to be resistant to certain diseases.
Yeah, they're also like modifying food to be grown in different regions. And the one that kind of pisses me off the most is they're simply using CRISPR to change the color of flowers. So you'll have different colored roses. I don't know if this is just an experiment or this is an actual business idea, but that kind of pisses me off where you have the literal hand of God to change something to anything you want and you simply change the color. That kind of pisses me off, Nick.
So we can finally get black tulips. Do you remember, did we talk about this when we were talking about? Okay.
No.
So the Dutch, I want to say.
Long ago in a distant galaxy far, far away.
But that's where all the tulip fields are, right?
Yes. And Nick, you know how I feel about the Dutch, right?
If there's two things I hate in this world, it's people who are intolerant of other cultures and the Dutch.
Oh, Austin Powers will forever live on.
So, but their tulips were a hot commodity. When I say hot commodity, like everyone grew their own tulips. People won prizes. People still do. People won prizes for the best tulips, and they were trying to change the colors. And the goal was a black tulip. And people would do all sorts of things, like put charcoal in the soil, like all of this stuff. There's a prize that some royal put out, a ton of money to whoever grew a black tulip. But all the tulips being grown everywhere led to disease that turned the tulips black and then killed them. So they did get the first black tulip, and everyone's like, what? This is insane. Like, they did it. And then the tulips all died because it was a disease. But this way, we could do black tulips without the tulips dying, ideally. So you could get the black tulip.
That is an interesting tidbit. I did not
There's
know.
a whole book written about, like, hypothetically, if a guy, like, he grew the black tulip, and it was like a murder, but it was like people murdered in the book. Like, they murdered the guy so they could steal it, and it was a whole thing. I mean, it was a craze.
I'll have to take your word for it. But also speaking of death, not the best transition I've ever had, but another big thing that they're trying to use CRISPR for is eliminating mosquitoes. Just simply getting rid of them or getting rid of the diseases they carry, which I don't know where they plan on using this, because if they plan on using it in the USA, a lot of the mosquitoes that carry diseases is because the U.S. government dropped mosquito bombs on its own self. So it's like, you better fucking fix your mistakes.
Well, we're trying. But yeah, I have heard about that, trying to get rid of the genetics of mosquitoes, allow them to carry certain diseases so that we can wipe them out. Getting rid of mosquitoes is not the answer, because you can't just remove one whole part of an ecosystem, however terrible they are. Trust me.
Sure we can.
If there's any insect I'd want off this earth, it's ticks. But they do play a role which would have far-reaching effects and wouldn't be ideal for the environment as much as I would love to never have to worry about getting Lyme disease, as someone who's picked multiple ticks off them this week because I work in the woods.
Well, it sounds like you need to get CRISPR, because with CRISPR, you might be able to cure Lyme disease.
No, true, but what I really want is that thing that cats and dogs have, where you just rub it on the back of your neck and you don't have to worry about it. I don't know how cats and dogs have that, but I don't.
Well, we can still put a collar on you just yet. But speaking of animals and something that they have that we don't, and sticking with CRISPR, I want to talk about telomeres, which...
Telomeres?
Telomeres? Yeah. Telomeres. I want to talk about telomeres because they are an interesting field, and it seems like a very interesting Venn diagram of telomeres overlapping with CRISPR and with cancer and with the animal kingdom, Nick. So with telomeres, those who aren't familiar, are pretty much the DNA's protecting coding. So yeah, that's the best explanation I could do, is they're protective coding. And they change with your age. So the shorter they are, the usually more aged you are, and the longer they are, usually the younger you are. But the longer the telomeres, the higher chance of cancer. So when you modify it, especially in mice get a lot of cancer. But what's really weird is that whales have long telomeres, and they don't get cancer. But with CRISPR, this could all change. So we might have the ability to change telomeres in humans, which would reverse the aging. And I think, if I remember correctly, the most we've reversed aged a human being was 10 years. But with CRISPR, we can do it we could do a decade, or we could do even longer, and use a CRISPR code to mitigate any cancers that pop up. So we might theoretically be able to reverse your age and make sure nothing bad happens to you once we reverse your age.
Well, that's the dream. That's the $1 million.
I thought it was to cure male baldness, Nick. I thought you were going to go with that joke.
Well, I mean, that's my dream. That's not the dream.
I don't know. I feel like that's also a multi-million-dollar process.
It's a probably $1 trillion industry right now. I can't imagine. But once you cure it, all those people are going to be looking for money somehow.
True. And I found it very interesting. I don't know if you came across a company called Loyal, Nick.
I did not.
Loyal, who was created by a startup company in Silicon Valley, started by Celine Haliula, which I probably butchered. I apologize. What you do is pretty impressive. But she started a biotech company, developed drugs to extend a dog's lifespan. And along with nanobots, she's also working with CRISPR to extend the dog's lifespan. This might sound minute, especially if you're not a dog owner, but one, you want your pets to live as long as you can. But two, dogs and humans aren't that different. So this might be a stepping stone of like, hey, let's figure out dogs, then we can just switch over to humans.
I mean, if you could extend a dog's life, I think people might even pay more than that for extending a human's life.
Yeah, I know. She's very interesting woman, and she's also our age, Nick, so that makes me feel sad that I have done far less with my life. But it's a very interesting concept. Start small with animal experimentations, which is extending animal life using CRISPR, and then maybe move on to humans. But that technology that she's using, Nick, me and you can order it right now. CRISPR is not this big thing where you need a bunch of permits from, not be a big company, need a lot of money. You can just order CRISPR and using some software, make what you want to make. Now, we can't guarantee it'll work, but it's based on you and your science. And I don't know if you want to switch into the backyard CRISPR.
Yeah, that seems like the perfect time to talk about CRISPR in the garage.
Well, the companies I'm talking about are like Odin or Gene Universal. Odin is a company which can sell an at-home CRISPR kit for about $200, and Gene Universal is a private gene synthesis company. And there are a dozen other programs like on SourceForge and stuff like that you can do CRISPR at home. Again, CRISPR founded all those patents in 2012. It's a relatively cheap and efficient. It's just how simple it is, but yet how complex it can do is absolutely amazing. And it's at the very edge of our fingerprints, at the very edge of our fingertips. And I want to get your initial reaction on it, Nick, about at-home CRISPR first.
So the other question is, how do I feel about people being able to use this? Because it is when I say relatively simple, I mean it's something people can do in their garage affordably. Not that means I understand it at all or that I could do it, but I am for and against it. I like the idea of people being able to experiment with it, but at the same time, it's terrifying because people can experiment with gene editing So I essentially have no opinion in all the opinions.
You're hot and you're cold and you're yes and you're no. I'm also torn, but my initial reaction is good. Let it be in the hands of the people rather than private institution. The main reason why, even though it is a dual-edged sword and could be dangerous, the main reason why I think it'd be better for the people to have is, again, you gotta use your brain power to be able to use it. Like, it's not like we say it's like copy and paste in a Word document, but it's a little bit more complicated than that. So there is a bit of a learning curve, which makes me feel like it's going to deter a lot of idiots who think they can just give themselves superhero powers. But at the same time, I have seen people turn their dogs to glow-in-the-dark dogs, I guess is the best way to describe it. Or like the founder of Odin, inject himself with a muscle-growing protein in the hope for bigger arms. It's mixed reviews, but I think the rewards out-compete the risks.
Yeah, and I guess, like you said, my point would be the more people experimenting, the more likely you are to have innovation, and you're more likely to advance this sooner and make it cheaper for everybody, which is why I'm in favor. But you can do a lot of crazy things with it, so it's kind of like, is this something I want everyone to be able to have? Yes and no. This is above, I guess, each person's going to have to make their decision, but I guess the matter is, it is out there and it is being done, so we're kind of past that point.
Well, we're not quite past that point because there are governments, and the U.S. government is one of them, trying to make it hard, if not impossible, for the everyday citizen to buy or use CRISPR, which I'm not quite sure how I feel about the government trying to regulate science. Especially, I understand they're trying to regulate backyard science, not, laboratory science, but if you're able to do it in your garage and able to, I don't know, it almost seems like more computing power if you're trying to solve a problem. Like, oh, you're trying to solve cancer? Have 5 million people instead of like 100,000 people trying to solve it. That seems a little bit more logical in my mind. But again, Nick, it only takes one crazy son of a bitch to ruin it for the rest of us.
Yeah, and that's the problem with super strict regulation, right? Is if we don't allow people to do it here, they're just going to go to other countries and do it with way less regulation, which is why I don't think an outright ban or super strict regulations is the answer.
Yeah, if they don't want to do it, if they can't do it here, they'll just go somewhere else.
Yeah, because there's countries who would be more than happy to have people doing this, and then we lose out here. Yeah, I think we need to have some incentive to encourage innovation in the United States. We shouldn't make it too hard to do business here. And this is kind of getting ahead of ourselves, but if you don't let people do it here, like I said, they're going to go somewhere else. And not only that, people are doing it in other places too. And so you don't want to lose out and be behind the curve because we just said, nope, we're not doing that.
Is it bad? I'm thinking at the same time making Captain America, but also at the same time I'm thinking of in China when they made the modified embryos and gave birth to the children.
I was going to say it's like making Captain America or making Homelander.
That's a much better analogy. And yes, that is, oh God, that one. Oh God. Yeah, that's a scary thought. That's a really scary thought. But I understand why there's some concern for regulations. One, not just the weapons of mass destruction, because you could possibly make a disease that can kill everyone, but also if you're making a disease with CRISPR, it's man-made. It's usually easier to cure than if Mother Nature makes it, because Mother Nature's a metal son of a bitch. So that's kind of a good sign. But with CRISPR, just in general, not even at home, that it's a light switch. So like I said, when you're fat or when you're eating healthy or if you're stressed or if you're at calm, you have different genes turn on and off. We're not quite sure with CRISPR if we implement that procedure on if we turn off, say, some part of your sequence of DNA that it turns off, and it might turn something else on that might be more even deadly. There's no undo button yet, Nick, no Control Z, but they are working on trying to have an undo button for CRISPR, which I thought was interesting. And probably one of the biggest ones I ever saw was CRISPR may pass on genetically, and I don't know if you want to exactly get into that.
I mean, I guess, yeah, if you change the genetics, you're going to pass on those genetics. I thought that was kind of understood. I mean, I guess I don't know what the because the idea I guess this ideally, speaking in broad terms, what we're going to do initially is turn off the bad genes, right? The stuff that allows you to get certain diseases. We assume, like, that's the goal. And the caveat to that is we don't exactly know what turning off those genes will do to a person. So if you turn off those genes, are you going to make them susceptible to something else? Are you going to fuck up something else in their life? And then those genes are going to be passed on. So I guess, if you're doing it in that sense, you're going from one fucked up situation to another. And I guess where the ethical dilemma is if you're doing like designer babies in a way where I want my child to not lose his hair. Well, changing that, does that have another effect that we don't know about? If it does, that's passed on. So I guess it gets ethically unclear when you start depending on what gene you're trying to, what physical trait you're trying to change the expression of.
Yes, I agree. But a couple things I want to add on is, one, I am scared that this is way down the line, like I'm talking another 5,000 years, humans become a monoculture like bananas. Two, most inherited diseases, I believe, are dormant, so you need both parents to have them in order for it to happen. So it's already a subdormant gene, and modifying a subdormant gene to be completely gone, what made humans advance so much was the mutations we have. And if we start removing mutations, I don't know if we're going to keep evolving or advancing. I mean, if you look over the past, 1,500 years, our brain technically shrunk but got more dense. Like, we're still evolving every single generation. And having that added non-natural DNA sequence in nature, it just seems like, one, we're doing away with Darwinism. Two, we are like Jurassic Park, meddling things we kept doing. We never thought, should we do it? And three, we don't know the after effects. So like maybe this is just worst case scenario, all hypothetical. This is probably fictional, but I still want to say it. I can imagine a parent trying to save their kid. They tell the physicians to use CRISPR to turn off or cut or change or silence a certain sequence of their DNA. Kid's healed. But because of that, kid lives on, passes on his genes, and meets another person with a specific disease, and the genomes just happen to match up to create a whole new disease. It's highly unlikely, but not improbable.
But that's always going to be a problem with mixing genetics. I mean, that's the con side, right? The pro is you mix two people who are, have comparable genetics or, good genetics that are resistant to diseases and pass it on.
Comparable? Are we in Alabama?
Well, aren't you like at some level, don't you find a mate who has what you're attracted to them for part of the reason that like you recognize they have traits that you don't have or genetics that you don't have that will create like I thought I heard something in like science class where you're attracted to the pheromones of someone who has genetics that will match up with yours in some way or another.
Not pheromones. Pheromones actually we look for similar pheromones, but we do look for different DNA. Humans like being mutts. We like mixing our DNA as much as possible. That is because much like dogs, kind of bring it full circle. A mutt is usually healthier than a pure breed because they mix DNA. They have usually the dominant genomes in each DNA, not the subdominant. So the diseases are less likely to occur.
Right. And so I guess I'm not as concerned about that because that's always going to be a risk when two genetics are paired. Could it be more of a risk when you're highlighting certain traits? Possibly, maybe even probably. But that's, I don't know. I'm just not super concerned about that.
I am. I don't know why, but the best analogy I can come up with in my mind is I'm imagining like a timeline. And as soon as you add a synthetic or a modified DNA, it changes the timeline. It's no longer a consistent evolutionary basis. It's a modified evolutionary basis. I'm not saying that we shouldn't do it, but I do say we should proceed with caution.
Right. So what comes to my mind to help explain this is you're talking about, random genetic breeding versus selected breeding. And when you select a breed for certain traits, sometimes certain traits show themselves in ways they wouldn't. So like if you're doing a crop let's say corn, and you're breeding for, you want it to be drought resistant and you want it to be produce good yield, but because of that, it might be more resistant to rust, and as time goes on, or whatever blight, whatever pest you're not as resistant to, now you have a lot of corn that's been bred for these certain traits which we didn't know at the time are more susceptible, and now that enough of these genetics are out there, whatever pest can run through them because the pest is stronger enough to overcome whatever resistance they had or lost through the breeding.
Yeah, that's kind of optimistic, but the pessimist in me is going with your Homelander analogy. We already are trying for the last multiple decades trying to make a super soldier. Why would the government, who has...
I mean, we have Tim Kennedy, Mike. I don't know what more do you want from us?
All right, touche. But we've already seen the CIA with LSD and the drug-induced programs that made tons of killers. Why would the CIA not mess around with CRISPR and try to make a super soldier or experiment on the general public without us knowing?
Oh, well, you just got to trust the CIA, Mike.
Got it. Yeah, I completely trust my government and all those overlords who rule over me. Thank you for letting me breathe another day. But another big thing is, genetically, I'm also thinking of what happens when we start having designer babies or designing creatures. Nick, I'll be honest with you. I can see people just deciding to change their skin pigmentation blue just for shits and giggles. And then genetically, they pass that on to their kids. That doesn't seem right to me.
Yeah. I mean, I agree. It's kind of like, it's like when parents let their little kids dye their hair like crazy colors. Like, this might just be an Oregon thing, but when you see parents who have like a 3 or 4-year-old with like crazy blue hair or pink hair, it's like, that kid's going to get made fun of. Especially when it's like a boy with like bright pink hair. It's like, what are you thinking? Like, you're setting them up for failure.
I was thinking of like the name, like their last name's like Kirschgoff. So you name them Josh. So when kids at school call him Josh Jerks Off, stuff like that. It's like when they name the names a little too easy to make fun of. Maybe do that. But like, I imagine people are going to do fads. I mean, look at tattoos. People to tattoo their entire body like a tiger. They metamorphic their body to look like an animal. They tattoo their entire body to be black. They're going to, using CRISPR, and probably not in the far distant future, change it so they might have webbing in their fingers. They might have, different colored eyes than anything ever seen in the kingdom. They might change the pigmentation of their skin. They might. I don't honestly know what's going to happen, but it doesn't seem fair that you're modifying your genome so your kids will have it even if it wasn't their choice. Like, it's one thing if it's your choice, but when you start forcing it upon future generations, that's when it kind of gets kind of iffy to me.
Right. And I'm glad you brought up fads, and this kind of ties in a little bit to the ethics here. It wasn't that long ago that people were discussing eugenics as a normal thing, which now we can all agree that that's not good. So eugenics was, the idea of breeding good genes together to create a better human race. And it wasn't that long ago that people thought that that was a good idea. I mean, the best example of that would be the Germany in World War II, but it's popped up around from time to time since then. And as enlightened as humans think we are, I guess the idea is that it's easy to get sucked into a fad like you were talking about. And when we start playing around with stuff like that there is a worry, I think it's a well-founded worry, that bad stuff could happen because we all bought into some fad.
Yeah, I'm just imagining like early 2000s when everyone had bedazzled jeans, but now just making your skin shiny like Twilight. That's a very petrifying thing. And I'm not saying that you should, but I can definitely see it happening. But your buddy passes out in the dormitory, so you think it's funny to stick him with a modified DNA RNA sequence so he has like bigger lips or something like that. That I can see absolutely fucking happening.
Yeah, I didn't think about that. That's kind of funny.
It's kind of funny until it actually happens, and it's like, oh fuck, like that belongs in a movie, not in reality.
Pick your friends wisely, I guess.
Very, very true.
But yeah, and then in going along with trends, the idea which is more concerning, not just the idea of eugenics, but when you start talking about made to order genetics, right? Like you're picking to lose or add certain things. The concern is that most people will choose to pick similar traits. Like everyone wants their kids to be healthy, but they also want their kids to be
6'2" and
strong.
ripped.
They also want them to be tall, wanted to have a good body. And the thought is that by doing these things, changing the genetics to achieve this ideal child, that we're all going to remove a lot of diversity in human genetics. The counter argument to that is that not every single person wants the same thing, and that whereas some people might be more worried about how intelligent their child is, or versus how physically big they are, and certain families want certain things, prioritize different things, because it's not going to be do you want your kid to be 6 feet tall or an idiot? It's going to be, do you want the expression to show, him to be more musically inclined or more mathematically inclined, or do you want him to be more athletic? We can express those genes, but it doesn't mean he's going to be those. I mean, once you start getting into those like behavioral characteristics and stuff like that, it's not as easy an expression as physical characteristics. But the fear with the physical characteristics is that people are going to be like, oh yeah, like you said, do you want like tall, strong, and that it's going to remove a lot of diversity we have. And I could see it going either way. I could see everyone falling into, like 60% of people all choose pretty similar genetics. Like we want our kids to be tall and strong, and we want, brown hair. We want, the good vision.
Or like Hitler wanted, blonde hair and blue eyes.
Yeah. And so, but I guess it's tough to gauge because I don't think I don't think you'll be able to pick and choose all of those. I think we can kind of hope for these set of genes tend to express this, but something could happen. I don't know. I don't know what I think would happen. I do think so I guess the idea is that modern quote unquote eugenics would be consumer driven based on what people want. And I do think that part of that is true, but at the same time, just think about like the targeted ads you're getting. Each of us is getting individualized targeted ads because everyone's has different interests and different ideals and different values. And so I think that there's a big enough difference in consumers that you wouldn't all get the same thing, but I think there's a big enough drive that most people would probably choose pretty similar traits. But I wouldn't bet on either or.
To add on to the fear, Nick, because I always love doing that, another concern of mine is when the government decides to, it's for the benefit, it's for the good of the people to CRISPR modify them so they have X, Y, and Z. No, we're doing it for your benefit. We're doing it for the good of your community. That's also a big concern of me. Because you have no longer control. I don't know why, but it worries me that someone else may have control over my DNA. Not just like a sample where they can plant it. I'm talking about UCIA. I'm talking about actually modifying who I am on a biological level is terrifying to me. But pinpointing from that is kind of the brighter side of CRISPR, besides curing diseases, which is absolutely wonderful. Something which kind of connects a lot of different things that we've talked about, Nick, and something you might enjoy because it involves plants. One of my favorite YouTubers, Thought Emporium, made spider silk using yeast. My first reaction was, one, that's fucking awesome. Second, why not use that as a manufacturing process? So, an example. Sunflowers are very good at pulling heavy metals from the ground. Why not modify the sunflowers using CRISPR so that all the heavy metals that they pull out of the ground all is located in one spot. So when you collect the sunflowers, it's easy to separate the metals from the flower, so you can literally grow metals. Or just like Thorium did, well, why not just create spider silk, which is a very strong natural material? Why not have entire yeast farms growing that, a new type of fabric to grow? Why not modify already pre-existing organisms to be more beneficial? Like, imagine modifying cow's milk to be more nutritious so less people starve to death. Why not, I don't know, have certain types of plants that grow different types of seasons? Like, you have an apple tree that blooms in the spring, the summer, and the fall. So you have fresh fruit all year round. You can easily do that with CRISPR. You can change entire construction and manufacturing industries. I'm trying to think of another example, like coral. Coral is kind of at danger, and it's actually not doing so well due to acidity and temperature in the ocean. But why don't we genetically modify coral to be resistant to that? To not only resist that, but to thrive on that. And the more coral, we keep them alive long enough, they'll eventually turn into limestone, which we can use. Or why not just have coral that has a shorter span that turns into limestone? So we can quite literally turn coral into an industry, a business. These are all easier said than done, but it has crossed my mind.
Well, I guess that's the problem people have with CRISPR, is that it can be used for pretty much anything, and it can take anything in the world to change it to how we want it. And that's what's exciting and terrifying about it, is that the possibilities are pretty much limited by imagination.
That excites me. I'll be honest. That statement alone excites me. Well, I got two more things for you, Nick. There's one thing I want to mention before this final question I have for you, unless you have any more questions for me, is back, again, 2012. 2012, I swear, Nick, the Mayans got it wrong with 2012. It wasn't the end of the world. It was the beginning of the end of the world. Because it seems like CRISPR comes around, everything happens in 2012. In 2012, Thorsten Staffenforst from the University of Tübingen in Germany found that a different kind of CRISPR method by linking enzymes and strands of RNA that could rewrite genes. And I want to point this out because CRISPR might not be the only thing that can be in eugenics, and might not be the only thing that can modify your DNA. There could be other things along the horizon. This is just the brightest star for right now. But my last question for you, Nick, is you have two answers. With CRISPR, say anything's possible with your imagination. Within reason. I don't expect you to be able to fly. What is your realistic CRISPR expectation for modification for your own body, and an unrealistic modification to your own body?
I think realistic would be immunity from either ticks or poison oak, and then unrealistic would be the ability to regenerate what's the tissue in your knees.
So you want to be the Wolverine, but specifically for your knees.
Yes.
That's not the worst. That's a pretty good one. I didn't think about that. Being the Wolverine is actually be pretty badass.
So what is your realistic? What's your unrealistic?
My realistic is fix my heart. My unrealistic, it would probably have to do with my cognitive ability. Like, be able to introduce photographic memory into bring that forward. Because there's got to be some gene turn on and off with memory that allows us to remember certain things, but to have the accessibility to back catalog quicker, easier, and better, that very much appeals to me. Almost like, you ever seen the movie Limitless with Bradley Cooper?
Yeah.
I imagine Limitless might not be a pill, it might be a needle with CRISPR in it. And that's probably my unrealistic one. But I'm not gonna lie, Nick, I still see all of them being somewhat possible.
I agree. So I had a little bit more I want to talk about. So we kind of touched on this with a little bit. So we talked about how in China they used CRISPR to affect humans, and how that works, and how the public perceived it as poorly. Humanity did not particularly care for using CRISPR to change genetics of humans quite yet. And I think that CRISPR is kind of like the next Manhattan Project, where you're damned if you do, damned if you don't, right? Because other countries are going to continue to research and invest and use this technology for good and bad. And if we don't continue to research and use this technology for good and bad, we won't know how to counter the bad. So I think we're at a point where we, as much as we don't like it, but we need to continue our research into CRISPR because everyone else is, and we need to stay ahead or on the curve. It was just kind of my thoughts on CRISPR overall, regardless of public opinion.
No, I agree with you. No matter if we agree with the policy or not, other people are going to do it. And for the very least, we should prepare for it. And what's the saying? If you want peace, prepare for war. It feels very similar to that.
Yeah, and that's pretty much the point. Is that we're past the point where we can just not do this. Everyone else has done it. They're going to continue to do it
at home.
Well, that's what I'm saying, and I think the government needs to start preparing for what that means globally. Whether that's how it's going to affect trade, how it's going to affect national defense. I mean, to a lesser extent, how it's going to affect culture. I mean, I don't think the government's going to be ahead of that, but at least on the defense phase, I think we need to start working on the possibility of something like this, some engineered virus coming out of somewhere.
Oh, yes.
Or whatever it is, whether it be, a certain country making super soldiers
or an organization gone rogue.
Yeah, Cobra, whatever.
I'm so happy you picked that up. Also, Nick, this is weird, but I'm also feeling a split in humanity because of CRISPR. Like, I'm not talking like we're going to have people who are fish mermaids, which is actually might be a possibility, but I'm talking like we may have two class of people, modified versus natural people, and I can see that being a very heated culture war of natty versus modified.
Well, that's the big fear too. Is initially this gene editing for for your children is only going to be available for rich people.
That's why I'm happy it's right now still available for the in-garage scientists, because that makes it shows how cheap it actually is, and it allows every person to at least have the opportunity to do it. But if it starts getting regulated, then, Nick, I'll be honest with you, I can only see the rich of the rich having access to it.
Right, and that's the fear that when only the rich have access to it, then they're going to use it, and then next generation, they're going to have a leg up, and eventually there's going to be, like you said, a split, depending on when patents run out and pricing and how that all works. You could see, like you're talking about, a split, and that's one of the big things socially that people are worried about is, how is this going to affect us long term in the future? And that's why people have discussion of, what traits are people going to select, and is it going to breed diversity, or is it going to breed
homogeneous?
Yes, homogeneous society. So, and I couldn't tell you either way, but it's definitely a concern that people have for good reason. But also, I don't want to be the first one to do it. So maybe let the rich people go first if they're so inclined. I mean, someone's got to try it out.
Well, the rich people aren't going to go first. They're going to have it experimented on poor people first. And then when it's safe, then make it exclusive only to themselves. That's what I imagine would actually happen. Not quite sure if it's actually true. But yeah, it's this is all very concerning, Nick. And I have, once again, no idea where the future's going. It's almost like a flip of a coin. Is it going to end today or will it end tomorrow? I'll flip a quarter and find out.
Well, that's all I had. So before we get out of here, Mike, what have you been reading?
I am still reading Geometry for Ocelots by Exterium. I have not had the opportunity to read at all this past 3 weeks, unfortunately. But hopefully I'll have more time next week. What about you? What book are you reading?
Still on the same page of Wood by Rolandinos, How Wood Built Society and Civilization. So
Nick, adulting is hard.
Yeah, sure is, buddy. Sure is.
And I do have a question for our listeners. What's the weirdest thing you can think of that CRISPR might be used for? And Nick, where could they tell us their answer to that question?
You can tell us on our Instagram and Reddit at Backyard Philosophy Podcast.
And who knows, if Elon Musk enters the DNA sequence of Twitter, that genome might change. See what I did there? It's my last pun. It's the last couple seconds of the podcast. I got to get one pun in.
All right, I'll allow it.
Well, that being said, thank you all for listening.
Thanks for listening to the Backyard Philosophy Podcast. We rarely finish a podcast without missing a point we wanted to bring up, so let us know what we forgot. And if you have a topic you want us to talk about, let us know at Backyard Philosophy on Instagram and Backyard Philosophy Podcast on